Hey folks, it's your friendly neighborhood AP biology teacher, Mr. Lang, and today I want to talk to you about mutations. Hmm. And the things that I want to talk about are different types of mutations, a little bit about how they arise, and why we care. So we've got up in the corner here, we have a little sheep, a little lamb, that appears to have at least one, two, three, four, five legs. Could that be the result of mutation? Absolutely. And oftentimes when you talk about mutations, that's the kind of thing that you think about, or like Teenage Mutant Ninja Turtles or any of that kind of stuff. But really, as you all know, that's not the end of the story. And mutations are really, really important for the evolution of all species and all life on Earth. So, for example, if I can get the slide to change, we've got these little rodents here called rock pocket mice, and they exist in all of these different color schemes. And if you were to find them in various places, you'll see that the ones that are best matched to the rock type or rock color are the ones that survive the best. And that makes sense evolutionarily, but the idea here is that those colorations had to derive from somewhere, and the place that they derive from is from changes in the DNA, which you also know. But we're going to put some names to various types of mutations as we go through the recording here. First question that's probably important to ask is a mutation somatic or is it a germline mutation? And the difference between those things is huge. So somatic means any cell other than the reproductive cells. Germline means only cells that are involved in reproduction. So if this fish experiences a somatic mutation, in order for that mutation to get passed along to its offspring, they would have to be well, it can't naturally pass it along. It would have to be a cloned fish. So there are some organisms that could pass it along that way, but anything that reproduces only sexually is not going to be able to pass that mutation along. If it's a germline mutation, that's a change in the DNA in either the egg producing cells or the sperm producing cells, and that is going to get passed along to offspring. So somatic versus germline, that's going to be really important when you're thinking about what kind of a mutation you're talking about, because one of them is likely to be passed along and one of them is unlikely to be passed along. And for some reason, this is not changing. There we go. So different types of mutations. The first kind of mutation that I want to talk about are point mutations. And another way to think about these is to call them substitutions, maybe. So here we've got our no mutation version. The DNA is TTC. The codon for that would be AAG, and that codes for lysine, an amino acid. The first type of point mutation I want to talk about is a silent mutation. So if we go to our codon wheel real quickly and think about AAG as being our codon, AAG is lysine. Well, if I go back here, if I go to AAA, AAA, it's also lysine. So in this case right here, this would be considered a silent mutation because effectively there is no difference between the original DNA and the new DNA other than the fact that we have one base different, but the outcome is the same. It's still lysine. Now, if I change something else, we go TTC in the DNA, then our mRNA is going to read AGG, AGG, that's arginine. That's a different amino acid. Remember, the primary sequence of amino acid is what determines the three-dimensional structure, that tertiary structure of the protein, and that determines its function. So if I put this in the wrong place, it could cause a problem. It also might not cause a problem. Maybe it just makes a slight variation in the coloration of the rock pocket mouse. Um, and maybe it helps it survive. Um, the third type that I want to talk about are nonsense mutations. And that is any time that you throw a stop codon. So if we jump up to the wheel, you'll see that in a couple of different places there are stop codons. Anytime you throw a stop codon in, it's going to stop the translation of the protein at the ribosomal level, and it's not going to do anything. The peptide's not going to be finished. When I say peptide, I mean protein. The protein's not going to be finished, and it's not going to work the way that it's supposed to work. So we have silent point mutations, we have missense point mutations, and we have nonsense point mutations. Those are important. Okay, we also can have frame shift mutations. And so in this case, if I was to, so here's our original DNA, CATTCA. They've skipped the step here of going to mRNA, and then they're just going directly to the amino acid language. So this is histidine, serine, histidine, valine, leucine, methionine, all the way down through. And what you end up seeing is that we've got that primary sequence of amino acids again. Well, if I was to get rid of this C right here, effectively they've done that and they've just shifted everything down. So if I delete this C, or if I added another C, that would be an insertion or a deletion, right? If I delete that C, I shift everything down, and you can see the primary sequence for this amino acid is completely different now. And if the primary sequence is different, that means the end structure of the protein is different. That means it's probably not going to do the job that it was initially intended to do and might not do any job at all. So frame shifts are a big deal. So if I add in one base, that moves everything down at the ribosomal level. It pushes it down one. If I move it two bases, it moves everything at the ribosomal level down two. But the most innocuous frame shift mutation, the one that does the least damage, would be anything in multiples of 3, because then I'm just going to be adding in whole amino acids worth of DNA. So if I add 3 more in right here, it's just gonna start. It's going to put one extra amino acid in, and then it's going to go right back to serine. The likelihood that one extra amino acid between these two is going to cause any major issues is not very large. Are there times where one amino acid change can cause a huge difference? Absolutely. Cystic fibrosis is an example of that. But for the most part, if I just throw 3 bases in, or 6 bases, or 9 bases, or 12 bases, the overall structure of the protein is probably going to survive. But if I take away 1, take away 2, take away 5, anything that's not a multiple of 3, or add in anything that's not a multiple of 3, there's probably going to be some issues. Also, the more you add in, the more likely it is that it's going to cause issues. That's frame shift mutations. Then we've got some other types of mutations. Let me move myself over. And these are all chromosomal. So here we've got a whole chromosome with sections A, B, C, D, E, and F, and you could assume that there's hundreds of genes contained within each of these. The first thing that you could do is you could just delete an entire portion of a chromosome. This is different than the deletion we were talking about here. Here we're talking about bases. Here we're talking about a whole section of a chromosome. That's a huge difference. So we could delete a section. So here they've just deleted section B. That could lead to all sorts of issues, because there's going to be hundreds to thousands of genes in section B that just aren't going to be able to be transcribed and translated into proteins. You can have a duplication, which can cause issues, because if the genes are there and the epigenetics are present to turn production of that gene on, if it's not on already, which it could be, then you're going to be making too much of a certain protein, and that can lead to lots of issues. We'll talk about a few different times that that becomes an issue later on. Inversion, we can flip flop parts of a chromosome and change things, and then also, oh, sorry, we could have this thing called a translocation, where we have a small piece of another chromosome break off and get inserted on this chromosome, which, depending on where it breaks, could be an issue as well. Another way that a lot of people like to think about mutations is by using something that we're already familiar with. And in this case, I want to use some words. So, so different types of variants when it comes to genetics, or different types of mutations. If my original sentence reads the gray cat ran down the hall, well, a missense mutation might just change one letter. Does it change the meaning of the sentence? Yep, it's missense. Okay, the gray cat ran down the ball. Doesn't make sense, but most of the gene is still there. Might even still do its job to some extent. It lets you know what the cat's doing. It's doing, it's doing something different than what you thought it was. An insertion, that's where we'd add a part, and that changes the gene as well. A deletion, now we don't know what we're even talking about because we've gotten rid of the cat, right? And then a duplication, the gray cat ran down the hall, which might be okay in the genetic scheme of things, and it might not. A nonsense mutation, that's when we throw in a stop codon partway through. We don't get the whole message. That's going to be a big issue. Hope this helps you understand mutations. Let me know if you have questions in class, and we'll see you soon.